spread by a number of different methods, both horizontally, person to person, or vertically, mother to fetus particularly. We break down those types of transmission into direct and indirect categories. As I said last time, those particular terms are not always intuitive because biological vectors involving things like insect bites are considered to be a direct method of transmission. I'm not sure. I can certainly understand why kissing, contact, for example, is direct transmission, but this one kind of baffles me a little bit. The indirect methods of transmission we tend to kind of underestimate and not be quite as aware of sometimes, particularly during winter flu and cold season. I mentioned that aerosols containing slimed bacteria and virus, for example, can be directly transmitted from one individual to another in close proximity. But those aerosols can linger in the room, suspended in the air, as droplet nuclei for several hours. And so you can walk into a room and inhale these particles even after the sneezer has left. And so that would be considered to be an indirect method of transmission. One of the reasons why colds and flu become more common in the winter is that we spend more time inside. Okay? That is one of the biggest factors involved. Now, in the past, I would have told you the old quote-unquote wives' tale that if you go outside and get cold, that can contribute to you getting a cold or flu was false. I was just reading some research the other day that's starting to question that a little bit, and there's some indications that lower temperatures in your nasal cavity, for example, interfere with your body's ability to produce some protective biochemicals like interferon, and that's particularly relevant with the rhinovirus that causes colds. And so, I'd always told students in the past there's no relationship between going out and getting a chill and coming down with a cold. And now it appears that there could be some connection. So we'll talk about that a little bit more when we talk about your immune response and how your immune response protects you in a number of ways. One of the things that we also tend to underestimate a lot are the risks of contamination of communicable disease from the food oral fecal route. Consequently, hand washing in the food industry is a very important part of disease prevention. And we're constantly in contact with fomites. Fomites are inanimate objects that are touched by people a lot, and as a consequence, disease agents can be transferred to those objects, and that if you touch them, you can pick up the disease agent. So what are some common fomites in your life? Some things that are touched a lot, might even be touched by other people. Doorknobs, for example. Computer keyboards, if you're using public computers. We used to say phones because people weren't carrying around their own cell phones, and if they were using phones, they were more public phones, and a lot of people have touched them. Most people don't let go of their cell phones and let people handle them that much anymore, so that's not as common of a fomite as it used to be. Another fomite that isn't as common as it used to be is money. Okay, money moving from hand to hand is constantly exposed to bacterial and viral agents. Did you know that paper money is impregnated with biochemicals to retard microbial growth? Okay, but we just don't use paper money that anymore. Okay, so as societal norms change, the fomites in your life become different, but they are still there. Another area where you run a great risk of infection is if you are hospitalized. About 10% of people who are hospitalized acquire an HAI. HAI stands for hospital-acquired infection. HAI is becoming kind of the preferred terminology right now for these infections. They used to be referred to as nosocomial infections. And I don't know why the name of the terminology is changing, but you'll hear in hospital settings HAIs much more commonly now than you will nosocomial. So, why are hospitals such a risk? Well, people bring microorganisms into the hospital environment. Sick people bring them. Well people bring them as well. Oftentimes people who are ill are immunocompromised. And because of the close proximity between people in a hospital, there's a very high rate of transmission of pathogen between staff and patients and among patients. And where those three circles come together, you have these hospital-acquired infections that develop. This is a significant public health problem. Somewhere around 2 to 4 million cases of HAIs per year nationally. About 100,000 deaths at a cost of billions of dollars per year of either direct costs or indirect costs resulting from extended hospital stays to treat these. One of the really scary parts about some of these hospital-acquired infections is that some of the pathogens are antibiotic resistant, and sometimes they are resistant to multiple forms of antibiotics. And so that's a pretty scary process. Hospitals have recognized the importance of prevention in trying to keep the number of HAIs at a minimum. And so typically there will be hospital committees which will monitor infection rates and also monitor compliance with certain conditions that have been established to try to prevent these infections. Probably the most effective method of prevention is hand washing. And if you've been in a hospital environment, if you were at JMH for your winter term, there are hand sanitizer things all over. Doctors and nurses, when they enter and leave the room, are supposed to use hand sanitizer and wash their hands. That direct skin-to-skin contact is one of the primary methods that you have of acquiring these hospital-acquired infections. Now, there are other ways you can acquire these as well. Catheters are a big source of HAIs. It is very difficult to keep catheters completely sterile. Fortunately, because of real conscious efforts, you don't get many HAIs anymore from surgery. We have sterile fields, we have antiseptics and disinfectants that are routinely used. But that direct contact between staff and patient, or patient and patient, is still our major source of problem in this situation. When we talk about disease, it's kind of appropriate to end up with a brief discussion of epidemiology. Epidemiology, ology are studies of, epidemiology is the study of the cause, distribution, and frequency of disease. In some respects, epidemiologists are kind of medical detectives. And when diseases pop up in the population, they ask questions like, who is being affected? What is the disease agent? How is it being transmitted? What is its pattern of transmission? Where's it coming from? A big part of epidemiology is tracing things back to the index case or the source case of that particular disease. There's a lot of different branches of epidemiology. Statistics are a very important part of epidemiology, as is data gathering. One of the big statistics that is routinely monitored and collected in epidemiological studies has to do with incidence and prevalence of disease. And it's important to distinguish between those two particular terms. Prevalence refers to the number of cases total in a population in a given time. So how many people are out there that are sick? Incidence refers to the number of new cases over a period of time. So prevalence, who is already sick. Incidence, who is just become sick. One of the things that epidemiologists do is they study changes in both incidence and prevalence of diseases over periods of time for different types of diseases in different populations in different geographic regions of both a country and the world. What they're trying to do is they're trying to get a sense of what the big picture is. And why do they do that? Because they hope that they can see patterns that will lead to treatment options and that also could lead to prevention programs. So until we established a clear link between smoking and lung cancer, it was really difficult to establish and get the public to accept smoking cessation programs. And so you've got to kind of figure out what the problem is, and particularly what the cause is, before you can then start developing programs to try to prevent this from happening in the first place. And obviously prevention is much more preferable than treatment when dealing with a particular disease. Now, there's a very, very well-established reporting system in the United States for gathering statistics and then funneling them to an agency that's kind of responsible for disseminating this particular information. And it all starts with the county health officers. How many of you worked with Dr. Mormon when you did your Johnson Memorial rotation? Anybody? Yeah, Dr. Mormon's been our local pediatrician for years. He was my kid's pediatrician. He is our county health officer. And there are certain diseases that have to be reported to the county health officer. By definition, by statute, these are considered to be reportable diseases. And here are the, right now, nationally notifiable or reportable infectious disease. You can just kind of take a look through here and see what some of these are. Some of these you probably heard of before. Others you probably have not heard of before. Okay, so polio is a reportable disease. Plague, we don't get many reports of plague. Various forms of hepatitis, measles, mumps. Some of these diseases are fairly rare, particularly those that we are immunizing against. But if someone comes into a doctor and it's determined that they have one of these particular diseases, that information has to go to your county health officer. County health officers then report on up the chain to the state Department of Public Health, and then states have to report their data of reportable diseases to the Center for Disease Control. The CDC in Atlanta also shares its information internationally with the World Health Organization, because particularly in our day and age now of pretty much instantaneous travel, diseases really don't know international boundaries. So it's important to know what's going on worldwide as well as what's happening in our country. The CDC publishes a monthly report called the MMWR. The Mortality and Morbidity Weekly Report. And this is intended to communicate to all physicians kind of what the trends are and what the tendencies are. And I've just excerpted a few pages of MMWRs to give you an idea of what kind of data is reported. It's really hard to see. I can't see this one unless I get even, right up there. So this is a report of selected notifiable diseases in the United States from October 13, 2012, from October 15, 2011. Pretty dry stuff. Here is a set of statistics and data from an MMWR that has to do with West Nile virus data, foodborne botulism data, and pertussis, which is whooping cough. So a physician, by going through this on a weekly basis, can kind of get an idea of what's happening nationally, what's happening in my area, and what kind of things should I be on the lookout for. Okay? So if we see a high case of something, West Nile virus in Illinois, for example, 252 reported cases in Illinois versus 23 in Indiana, that's useful for a doctor to know. That's useful for a doctor to be on the lookout for, because West Nile isn't that common, and it's something you might miss otherwise. Okay, here's some HIV data from 2017, estimated number of HIV cases. TB cases by age group and sex in the United States in 2008. So you get the idea. This is useful information. Typically, as we monitor the change in incidence and prevalence of particular diseases over periods of time, some trends start to develop and some patterns start to develop. One disease pattern is to say that a disease is endemic. Well, this is another one of my slides. I'm trying to figure out, the images disappeared on these slides, so I don't have those. I'm going to try to find those and replace them when I put them on Moodle. A disease is considered to be endemic if there are a constant number of cases in a given region over time. Typically, the number of cases is relatively few. So Lyme disease, for example, used to be endemic to pretty much the New England states. But because of a number of environmental factors that have expanded the range of the white-tailed deer, which is the normal host of that particular bacteria that causes Lyme disease, the geographic spread of Lyme disease has occurred over the last few decades, and now it is also endemic to Indiana. And so patterns do shift over time, but endemic refers to a particular pattern in a particular region, fairly constant in nature. A disease is considered to be sporadic if there are just a few cases here and there. And there's no real geographic rhyme or reason to where they're popping up. Rabies tends to be sporadic. You'll see one report of a case of rabies in one state, then you won't see it for years. And then you'll see a few cases in a totally different state, and you won't see it for years. Epidemic. The term epidemic is typically kind of misused by the general public. What does epidemic imply to you? What do you think of when we say a disease is an epidemic? A lot of people are sick, right? And so typically we associate that with high numbers of infections. But the absolute value of the number of people infected isn't a criteria for something becoming epidemic. For something to be epidemic, the number of cases has to exceed normal patterns or normal expectations. So if all of a sudden we see a jump in the number of cases versus what the historical pattern has been, that is considered to be epidemic. For one particular disease, it might be thousands of cases. For another disease, it might be hundreds of thousands of cases. So it really doesn't have to do with absolute value of number of people sick. It has to do with, are you exceeding the statistical expectations that normally you have for that particular disease pattern. A pandemic is a worldwide epidemic. So typically we use the term epidemic for a particular geographic region or country. So in the 1920s there was a worldwide influenza pandemic, the Spanish flu, that killed like 20 million people. Killed more people than had died in all of the wars at that particular time. It crossed geographic boundaries. And given the ease of air travel right now, public health officials are very worried about diseases like influenza, which normally have their origin in Far Eastern countries, spreading to other countries and becoming a worldwide pandemic. Epidemiology is a pretty complicated field. And epidemiological studies are typically of three types: descriptive, experimental, and analytical. Descriptive has to do with kind of what's out there. Okay, what are the diseases? What's their location? What kind of patients are being infected? A big part of that descriptive epidemiology is trying to trace the disease back to its origin. So to find that patient zero. When we talked about the history of microbiology, I mentioned John Snow, who's considered to be kind of the father of descriptive epidemiology. And he was able to collect data by going door to door and figuring out who had cholera and who didn't, and then mapping those particular cholera clusters, and as a result, finding that the clusters were all centered around that contaminated water pump in the center of town that was the source of the cholera bacteria. So all of that descriptive data that's being collected is useful in trying to find source. And that can be very difficult to find sometimes, particularly in foodborne illnesses. So if we have an outbreak of foodborne hepatitis, for example, you have to interview all the people and say, where have you eaten in the last month? Well, that's pretty hard for people to remember, and as much as we depend upon fast food and eating out, it can be really difficult to kind of trace that back and find the common denominator that the people have. Once a disease is kind of identified, then the analytical types of studies kind of study that disease in more detail. How's it being transmitted? And particularly, does the mode of transmission suggest possible prevention plans? There was a lot of work done in analytical epidemiology with AIDS. Okay? Once the pattern was determined, and it was determined that there were groups of people that were at high risk for AIDS, then that analytical work of how was it transmitted, what was the nature of the virus, and finding out that it was a sexually transmitted disease led to a lot of really effective prevention programs, including use of condoms. Okay? But we had to know what we were dealing with first, what the nature of the disease was, and how it was spreading before those particular public health programs could be adopted and be effective. Experimental does a lot of hypothesis testing. It's very hypothesis driven, and it relies a lot on statistical analysis of your experiments. This is where Koch's postulates definitely come into play. Remember, Koch's postulates are the gold standard for determining the cause of particular diseases, what pathogen causes a particular disease. So if you want to get into epidemiology, there's a lot of different avenues that appeal to a lot of different kind of personalities and traits. Mathematic abilities, pure science abilities, more kind of sociology, kind of public interest ideas. And so epidemiology is a very interesting career, and one that has a lot of important impact on our public health system, particularly for developing disease prevention and treatment programs. Okay, so that's some information about kind of the offensive side of pathogens' ability to cause disease. So what are some of the offensive weapons? Now it's time to take a look at defense. We're going to spend much of the rest of the semester talking about the human immune system. It's really fortunate we have a robust immune system, because hopefully you have acquired in this class an appreciation for how many different microorganisms you are exposed to out there in the real world. And not all of them are pathogens, but those that are pathogens can have some pretty impressive adaptations, virulence factors, that allow them to invade your body, colonize your tissues, proliferate, and make you sick. And given the number of things you are exposed to, it's really kind of amazing that we're not sick all the time. And that really is a testament to the immune system. The immune system is a complicated process. Some of the characteristics associated with this particular system is that it can have a very high level of specificity. Certain levels of it are highly adaptive. Certain levels of your immune system have a memory. I've already made reference to that. The immune system is highly redundant. Does anybody know what that term refers to when we say a system is redundant? United States space program has been a pretty safe program. Give a few real disasters. One of the reasons that space program has been so highly effective is that it is highly redundant. That's kind of one of the philosophies. What redundancy means is that if you have a system, there's always a backup, and for that backup, there's probably another backup for the backup. So there's a lot of backups involved in the way that this system is designed. There's a lot of overlap, if you like, and several different ways to accomplish the same thing. Another thing that characterizes the immune system is feedback. And the feedback built into the immune system can both be positive feedback and negative feedback. Now remember what positive feedback involves. Positive feedback involves the system kind of stimulating itself. So positive feedback is kind of amplification. Well, when your immune response is just starting to gear up, amplification is really important. But do you want your immune system to just keep raging out of control? No. At some particular point in time when the threat has been eliminated, we need to have the immune system shut down. And so negative feedback is oftentimes involved in modulation or self-correcting and turning off the response when it's time to turn off the response. There is a tremendous amount of cell interactions involved in the immune response. And a whole lot of chemical signaling that's involved. A big part of this chemical signaling is receptor recognition. So you want to make sure that the right cells are talking to the right cells chemically to have a good coordinated immune response. I'll be honest with you, when I start mapping some of this stuff out, some of the diagrams that we have up here on the board get pretty confusing looking, in part because of all of this redundancy, feedback. Probably can harken back, if you took animal physiology, to some of the feedback mechanisms that Dr. Rhodes would show you about how things self-regulated in the body. Well, some of these diagrams that we'll end up with going through the immune system look even more complicated than some of those that you thought probably were pretty horrendous. So this is material that you've got to keep up on. This is material that probably isn't that intuitive to you, and there's a lot of terminology associated with this that you probably have not heard before. So let's take a look at kind of a big picture look at what's happening, generally speaking, in your immune response. Ultimately, the goal of the immune response is eliminate foreign threats. And there's a lot of different potential pathogenic threats that the immune system has to deal with. Bacteria, fungi, viruses, protozoa. But the key here is foreign. Okay? Ideally, the immune response reacts to these things that shouldn't be in your body, but leaves the cells that are a normal part of your body alone. Now, sometimes that doesn't work, and we develop diseases that are the result of autoimmunity, where your immune system cells attack your own tissues, and that's problematic, obviously. Well, how does your body recognize these particular foreign threats as being foreign, as not belonging? Well, their cell surfaces look different. They have different molecules on their surfaces. And many of these molecules that are different are antigenic. Remember what that means. An antigen is a molecule that stimulates the formation of an antibody. So because these pathogens have foreign antigens, as opposed to the self-antigens on your cell, the immune system can recognize them and respond. So what if your immune system does get geared up against a foreign threat? What if it detects foreign antigens on cell surfaces? What can it do? Well, your immune system can produce protective molecules called antibodies. And what do the antibodies do? They bind to the antigens. And that binding is very specific. The specificity is much like the specificity between an enzyme and a substrate. So it's got to be complementary. What happens when an antibody binds to an antigen? It marks that antigen for elimination. It tags it. A lot of people have the misconception about antibodies that the antibodies themselves are some kind of killing molecule that directly destroys the pathogen. That's typically not how it works. Instead, the antibody is a marker that says, this is foreign. And then that is recognized by other immune cells like phagocytic cells. And the phagocytic cells gobble up the foreign antigen that has been marked for destruction. So the phagocytes are actually the killers. It's kind of like the old classic, junior high joke. Putting the kick me sign on someone's back, and as they walk down the hall, they get kicked. Okay, the kick me signs are the antibodies that target you for abuse. And then the kicking is actually the phagocytes that are actually doing the elimination. This part of the immune response is called the humoral response. And it has nothing to do with being funny. Humor is a Latin word for fluids. So these antibodies, when they're produced, are secreted into the blood and especially into the lymphatic system. And that's where a lot of this binding occurs between the invader and the antibody, and that's where a lot of the destruction takes place, particularly in the lymphatic system. One of the primary types of cells that are involved in this humoral response is a type of cell called a B lymphocyte. They're the ones that actually produce the antibody. If we start digging into this, we're going to see that there are other immune cells that are involved in this humoral response as well. But the ones that actually produce the antibody are the B lymphocytes. There's another way to eliminate foreign things if they are detected by the immune system. There's another type of immunity called cell-mediated immunity. Cell-mediated immunity involves a different class of immune cells called T lymphocytes. Two major types of T lymphocytes: killers and helpers. What are the helpers helping? They're helping the B lymphocytes make antibody. So even they are considered to be part of this cell-mediated immunity. They have a very important role that they play in this humoral response as well. What do the killers do? They kill your cells. You go, whoa, wait a minute, that must be bad. That must be kind of autoimmunity. No, this is good. Give me an example where it would be good to kill your cells in your body if they become cancerous. And what's one of the things that happens to cancer cells? Typically, the cancer cells' surface molecules, antigens, change as a result of being transformed into a cancer cell. And as a result of that, normally what your immune response does is it identifies that cancer cell as being foreign and results in an activation of the cell-mediated response against it. What's another situation where it might be good to sacrifice some of your cells in your body? If they become infected by a virus. Because what happens when cells become infected by a virus? They turn into virus factories. They lyse and they release more virus particles. So if you can kill the virus-infected cells early on in the infection, you can short-circuit them. One of the big hallmarks of your body's immune response is surveillance. And there are cells in your body that are constantly moving around within your tissues, looking for things that don't belong, looking for those things that are considered to be foreign, and then if they detect them, mounting a response against those foreign things to hopefully result in their elimination. So I've gone ahead and posted this next set of materials now and some PowerPoint slides for it's titled Line 1 and Line 2 Defenses. Are up right now, lecture notes are up and study guides are up, and we will continue with this on Thursday. So again, don't forget, make sure you've got a good slant for your unknown for Wednesday, and we'll do a bunch of fermentation tests. Oh, let me dig some out and find some. So you can just do that in lab tomorrow. Sure.
It almost like the